this and some of the features of bacterial genomes, just so that you're sort of all on board with some things about bacterial genomes. They're generally circular. They have minimal introns and non-coding sequences. Most of their sequence actually does something. Either it forms into an RNA or it codes for a protein. They have between 1 and 2 chromosomes. And it's only one group that actually has 2 chromosomes. They're called Berkeley, and they're a really, really nasty pathogen in Southeast Asia primarily. They don't have organelles, but a lot of them do have plasmids. And those plasmids are a source of some of the nastier things that bacteria tend to do. So if you've heard of hamburger strains of E. coli, so the nasty one, when you open the paper and you read something about Jack in the Box, having had some sort of nasty outbreak and 32 kids like getting bad Happy Meals, what happened was a regular E. coli, which is everywhere, E. coli in small amounts on any surface, you'll find it, picked up a plasmid. Somehow it transferred a plasmid from a really bad E. coli to this guy, or it got it from another kind of bacteria. Bacteria are fun in that they exchange DNA. They talk in DNA at times. And so they've gotten a plasmid from a nasty pathogen, and they have in turn become a nasty pathogen. So bacterial genomes, relatively small in genome size and genome count, and they typically have what are called Shine-Delgado ribosomal binding sites, which are the way in which genes know where to attach. So where to attach on the genome to actually form genes. Actually, Carsonella don't have it. Don't have Shine-Delgado sequences, and I don't know of another bacterial organism that doesn't. So once again, another fun feature of this gene.
A lot of genes in other organisms don't have Shine-Delgado.
Yeah.
Is that because of its symbiotic nature, or?
It's probably because it has what's called translational coupling, which is where the start of a gene and the stop of the next gene overlap. Yeah.
What's the relation between genomes and gene? Those two words I don't understand.
Oh, great.
How
Yeah. So a genome is just the total collection of genes in an organism. It's total amount of genes, and so each gene is actually located physically on a structure called a genome. And so it's just a physical structure. People have chromosomes, and those chromosomes are just a package of genes. And so your genome is your collection of chromosomes, and your chromosome is your collection of genes. So bacteria only have 1 to 2 chromosomes and some plasmids. So their genome is their set of chromosomes and plasmids, similar to us.
So where's the chromosome here?
The chromosome here is this guy.
So
Inside this organism, it has one set of this sort of all wound up somewhere. It's not as perfectly circular as that one. It's actually in the organism.
So where does the double helix come from?
So the double helix, you see these orange ones and these red ones? So I actually made this figure, and the reason is these are genes that are on one particular side of the helix. If it's red one way. So if you go up, so you have a set of base pairs, which are your A, G, C, T. And on the other side are an exact complement of those, so that your A's are forming with G's, and your C's are forming with T's. So everywhere where you have an A on one side of the helix, you have a G on the other side. And so genes are read in one direction on one side, and in another direction on the other side. So you can store data on both sides of the genome.
Just to throw out some jargon briefly, are there concepts like promoters and epigenetics and methylation and something like that?
So you definitely have promoters in bacterial genomes, which are just parts of sequence leading up to a gene that say that this gene is going to be active. It gets it started. It turns it on or it turns it off. Usually in bacteria, it turns it off because most of these are already active. Most genes are active on their own. Epigenetics and methylation are not as important in bacteria as they are in eukaryotes. So in multicellular organisms, epigenetics is really important because they have sort of a sense of offspring that is different from the sense of offspring in a single cell organism. And so you carry your maternal epigenetics with you when you're born. I mean, that's the sort of stuff that's in sort of New York Times Science every few months, is that, you know, your great grandmother smoked and now, you know, you have asthma. And so this all has to do with features that are emerging maternally, but bacteria just split. They don't, or sporulate, which is they just sort of pop off a little end of themselves that turns into yeah.
But it still have some of the epigenetic mechanisms, right? The methylation and so on. That probably still has to play a role here too. You know, wouldn't it? Because, you know, every time they go through some mitotic subdivision, right? So the DNA has to unfold. I mean, there's all these processes that control that, right? So wouldn't there be any role for those kind of processes?
Actually, I don't know that much about, yeah.
Oh, actually, well, for epigenetics, it applies mainly to shape of the DNA into compact into chromatin. Yeah. And then in chromatins, they have like special chemical groups attached to them. And then these, depending on which site they're methylated, it depends like which part will be expressed and such.
But even here, shouldn't such mechanisms, that's my question, right?
Oh. Well, like you said, it mainly applies to multicellular organisms because the more bigger and complex their genomes are, the more regulation you have to add. So that's why you have epigenetics and methylation. And also you have the chromatin packing that helps it compress it.
Oh, I see. It's not that sophisticated.
Oh, no, it's definitely not.
Right.
It's like comparing a ball of dirt with a single grain of dirt. Got it. That kind of thing.
So bacterial chromosomes don't tend to supercoil the same way that eukaryotic chromosomes do. So you have a lot more genes even in an individual chromosome, and so they have to be on the surface somewhere to get.
And methylation in bacteria is supposed to be related to restriction enzyme sort of defenses. It's not really epigenetic.
Yeah.
The way
I see. Okay.
It's not genetic. It doesn't restrict translation or transcription the way that it does in other organisms. That I know of, right? I mean, no one's really.
Question.
Yeah.
It's going back to that you can read DNA in both directions. So can you get useful proteins in both directions as well?
Yes. But they won't overlap by and large, at least not by more than about 3 base pairs. So by about 3 nucleotides is the max in which they'll still be functional and overlapping. By and large, these are completely staggered. In a genome that's filled, so it doesn't have much in terms of inner gene space, so it doesn't have much non-coding. You'll just see them sort of right there. It'll be the start of one, or the end of one, the start of the other. And then if it's in the opposite direction, it's the opposite. So the start of one, the start, end of the other. Yep. Those are really good questions, guys. So in terms of genome size and gene count, bacteria are low, but nowhere near as low as viruses. So a lot of viruses maybe have like 5 genes or 15 genes, and that's really all they need. Viruses are a much different kind of organism than a bacteria, though. But bacteria are very dissimilar to eukaryotes and sort of where they hit on this map right here. So right here we got gene count, 10 to 100,000 genes right there. And then we have genome size, about kilobase, so 1,000 base pairs to about 10 billion base pairs. A lot of these ones on this far end are plants, almost exclusively plants. So the biggest genome out there is, I think, the lolly pine. It just came out. It's like 21 billion. It's huge. Humans are 5, I think. I'm sorry.
3 billion now.
Three, yeah.
I used to be 5.
Yeah.
There also used to be 70 or 100,000 genes, and now there's like 23. 20,000. These things change.
Yeah.
Like Carsonella, like the subject of today is. Oh, that's really all the
Small.
viral genomes on.
That's a great map.
To give you a sense of like how outgrouped we are right now.
We're in viruses.
Yeah, we're in the big virus area.
It's interesting that this map is almost linear, right? You can see there's some stuff below the line, right? Below the line.
Yeah. So especially in terms of gene count, but this is an underrepresentation because it relies on us being able to count genes incredibly well. So this number, no matter what, this is an underrepresentation of the number of genes, probably.
The slope's really higher.
The slope's a little bit higher.
But on the bottom end, there's so few genes that it just won't. I mean, it just doesn't behave the same.
And so this is not, with so many data points, not a heavily curated list. So this isn't all the best genomes that we know everything about. This is from 2001. It's Kunin's data. And so we're better now than this data is, but this is the best collection I know of these. So there are a few really small genomes. Mycoplasma genitalium, which is about 580 kilobase. It's 580,000 base pairs, 525 genes. And then we have the groups. So this is the smallest free living bacterium. So it can live on its own. It doesn't have to exist with some other host. These guys, Buchnera, Carsonella, Tremblaya, and these ones all live in the guts of sap-sucking insects. So we'll focus on Carsonella in particular.
All 3 of those, the smallest ones.
Yeah.
Interesting.
And they're all over the map in terms of their evolutionary history. So these are not like one small group. These are all over the place. Figuring out exactly where is its own kind of problem, but we do know that they're all over the place.
Kind of interesting, on the full other end of the spectrum, the lolly pine has sap, right?
Yeah.
And it has the most number. And so it's interesting that wide divergent. And yet there's interrelationship here.
So it's interesting, the relationship that goes from getting all the way from plants to these bacteria because you have a whole bunch of specializations that happen. There's an insect that only feeds on a particular plant,
Right.
and a bacteria that only feeds on a particular insect. So you have this super duper specialization of it happening, and that specialization resulting in this extreme reduction in genome size. So to get a sense, just in another bacterium, probably our best known bacterium, well, definitely our best known bacterium, E. coli, we're talking about 5,000, 5,400 kb versus 150 kb. Huge change, and both of these are, single chromosome. This is the chromosome size, not the genome size, so that's for one chromosome.